Okay, welcome everybody to this next IPC Partners Journal Club. Good afternoon, good morning, good evening, depending on where you are in the world. Delighted today to have Dr. Mark Garvey as a special guest on the Journal Club. For those of you who don't know Mark is Director of Infection Prevention Control at University Hospitals Birmingham in the Midlands in England, and also Director of the Hospital Infection Research Lab. So a big welcome to Mark. Just before I pass over to Mark, I'm just going to do our usual housekeeping that we always do. It's a bit of a celebration actually for John and I. Mark, this is our 10th Journal Club, believe it or not. So we're in the double figures. For those of you who haven't joined before, we've just got a bit of an introduction about what IPC Journal Club is designed to do. So usually John or I or a guest speaker will talk about an insight into a topical IPC published paper. And today Mark's going to be talking about his publication in Journal of Hospital Infection entitled The Splash Zone. Really though, we'd love it to generate discussion, and we've been really encouraged and enthused by the question and answer part of these journal clubs, which usually comes after the speaker's finished. So we'll have plenty of time for Q&A. There's a question and answer box in the top, so please distinguish between Q&A and chat. So please use the Q&A for any questions that you might have for Mark, and John and I will deal with them at the end. And hopefully we're building a bit of a network of enthusiasts as well, to have a little bit of fun. And we'll do a wrap up at the end, but just to say that all these sessions are recorded, and they'll be posted on our YouTube channel for IPC Partners, which you can find on YouTube. So we'll stop sharing now, and then I'll hand you over to Mark. So Mark, over to you, and thanks again for coming on today.
The pleasure. So hopefully, is that shared for you guys? Yeah.
Yeah, we can see that.
Thank you, Mark.
Brilliant. Let's hopefully that will enjoy. So yeah, thank you for asking me to talk today. So I'm going to talk about the sink splash zone. Giving it what's lurking in your taps and sinks. So I've given a little bit of an overview. What I'm going to talk about is a little bit of how we got to that point of why we wanted to look at the sink splash zone, and a little bit of work afterwards. So it's only a few slides to begin with, so hopefully those are working. So when I first started at UHB many moons ago, one of my first interests was in Pseudomonas aeruginosa. So anyone who knows me, my favorite bug randomly is, well, not randomly, is Pseudomonas aeruginosa. There's lots of reasons why it's my favorite bug. Number one is basically how it smells. What's a very weird thing, really. But a few years when I joined the Queen Elizabeth, we had a bit of an outbreak of Pseudomonas aeruginosa in one of our critical care units. So anyone who's heard me talk before, I always talk about our critical care unit. It's like 100-bedded. A single 100-bedded co-located critical care unit. What's massive really when you look at it. And in one particular room, we had 5 patients who had Pseudomonas aeruginosa. And when we did some typing and sequencing, we found that they all had the same strain. So this is like a burn shock room that we have on one of our critical care units, and they all had the same strain. Now, when they're admitted, okay, they're burns patients, extensive big burns patients, these would be, none of them had like Pseudomonas aeruginosa when they were admitted, and each single patient who went through this room actually had Pseudomonas in a burn swab, and it was the same type. So when we had a look at it in a bit more detail, we actually found the same Pseudomonas aeruginosa in the water, in the sink tap. In that room, we obviously followed the addendum, did certain types of remedial works, and again, like 6 months later, we found the same strain. So we reported this, talked about it, published it, and then that led us on to some more work. So we were lucky enough to get some funding from the Department of Health to look at the role of Pseudomonas aeruginosa transmission in an adult augmented care area. So we know that when babies are born, we had all those neonatal outbreaks back in 2010, 2011 in Bristol and Belfast, where babies, when they're born, they're not colonised with Pseudomonas, and then we found infections with Pseudomonas in these neonates, and it was linked to the water. So we wanted to do look at what the role is of Pseudomonas in an adult population, because adults can, have Pseudomonas on them. So what we did, we got a bit of funding to enable us to take some water sampling of our water outlets on our ITU and do some whole genome sequencing of the clinical isolates of our patient clinical isolates, and if we found Pseudomonas in the water, of our water isolates. So it's a big whole genome sequencing project. So it was the largest whole genome sequencing project for Pseudomonas in the world at the time. So we've got loads of outlets on our critical care unit. We've got 231 outlets, what's massive. And to our surprise, we found about 30% of those tap outlets at any one time. We can find Pseudomonas aeruginosa in them. What's quite scary, we have lots of patients with clinical isolates as well. But the take-home message from this study was, this is the dendrogram on the right hand side, where we've had lots of diverse strains, but what we can show is we definitely get transmission, very similar strains, exactly the same strains of Pseudomonas in our water to our patients, and we know we get transmission of Pseudomonas aeruginosa to our patients, and we had up to like 60% transmission. So it's a lot of transmission when you look at it. We obviously didn't leave it like that, and we did follow-up work. Well, although this was published a little bit before that whole genome sequencing project, we did lots of different interventions to try and reduce down the amount of Pseudomonas aeruginosa infections that we have on our intensive care unit. And we did various different interventions. I'm not going to go through these in massive details, but like on the right-hand side, it shows you a graph of the rate of Pseudomonas aeruginosa infections in our critical care. And this is just a breakpoint analysis, but number A is basically the burns outbreak that I showed you at the beginning, not burns, but the burns shock room outbreak. And we put filters on there, and we saw a little reduction down in Pseudomonas aeruginosa infections. And B and C were like holistic interventions, looking at how you dispose of patient wastewater down the sink and how you clean the taps, things like that. And then finally, D was like an engineering one, looking at different taps. So you can show that by doing different interventions, you can reduce down Pseudomonas aeruginosa infections. And there's been lots of studies looking at not just Pseudomonas, but other gram-negatives as well, and similar interventions can reduce everything down. We then followed that up and looked at like what other areas are in our augmented care areas do we find transmission of Pseudomonas aeruginosa, and we found it on a haematology ward. People will have heard me talk about this before, but we've had like mini outbreaks, and we've seen transmission to our patients in our haematology ward. So this paper was looking at, again, more holistic factors, but we had 3 patients who had an infection with Pseudomonas aeruginosa, and they were in the same side, well, not the same side rooms, but they were like, one was in one side room, and the next side room to that patient had Pseudomonas aeruginosa, and the next one had it to that. Now it was a bit weird, because I thought, well, where have they got this Pseudomonas aeruginosa from? Because we've got filters on all the taps, but in our IV prep room, that was the only tap that didn't actually have a filter on. And when we took water from that tap, we found Pseudomonas aeruginosa. But what also we found like these trays next to the in the sink and next to that tap outlets, and we could culture Pseudomonas aeruginosa the same type as the patients in that. So we know we get transmission of Pseudomonas that way as well. And that's just one example, but there's other examples. This is a complicated piece of work we did with the university, but we found Pseudomonas aeruginosa transmission similar to our augmented IU work. We found transmission to our patients in our burns population as well. So these are difficult diagram, but it's one that the university devised, but it's simple when you think about it. But see, if you take box 4, for example, the different colours represent like water samples. That is for blue, red is for wound swabs, Eve's from the environment. So that patient in 4 had a Pseudomonas that was similar to the water sample in the environment. So we know we find Pseudomonas in the environment, and we can find it in the water. So then that led us on to looking at a bit more detail into sinks and what's down your sinks and things like that. And you can get, there's loads of articles, but these are scary. These are horrible pictures when you look at it. So these are some of ours as well. What is down your sink and drain is quite frightening when you look at it. And there's lots of details of outbreaks from like sink drains and what have you. I quite like this piece of, Kathy Noakes once showed this, but this is actually a water droplet, so hopefully that shows. So that's just a water droplet dropping down. Can you see all those little, like, well, hopefully you can see all those little sprays. After that, there's like microscopic particles from that water. And when I saw this, I thought, oh, I wonder how far water splashes from a sink. So just imagine if that water droplet had Pseudomonas aeruginosa in, just look at all that Pseudomonas that drops and sprays everywhere, what potentially you could have. Or if you had splashed onto a sink and what have you, and you had bugs in there, look how far these water droplets could go. So that led us to the paper that I'm going to talk about very briefly, was the sink splash zone that we published in a couple of years ago. And it's a really crude piece of work. All we did was thinking, okay, I wonder how far water does splash. So we put paper towels on the floor and ran the tap and see how far water would splash. And we found it could splash up to 2 meters away from the tap. So this room actually is our burns shock room. So you remember that outbreak I talked at the very beginning. This is the same room that we had that those cases of Pseudomonas aeruginosa, and this tap was actually the tap that had Pseudomonas aeruginosa in. So you can see how far it can splash up, and the patients can't see the patient's bed, but the patient's bed would be there. And actually, when you wash your hands and do different interventions as well, you can get more and more splashes from around there. And then what the paper goes into, we actually did an audit. So Alison Gardner from my team did an audit to see what kind of equipment was around the splash zone. And this is a typical critical care bed space. And you can see there's lots of equipment around there, even the patient beds within that sink. You know, you've got an IV trolley, you've got your PPE, you've got your bins, you've got your computers on wheels, the patient themselves. So there's loads of like equipment that's around there. And this is just what's presented in the paper, but Ali's done some nice posters of it. But you can see the vast majority of equipment. What shocked me, what's within 2 meters of a sink, everything from IV access devices, ventilators, dialysis equipment, personal care items, enteral equipment, your PPE, your alcohol hand rub, cleaning equipment, care devices. The list is endless when you look at it. And it varies what percentage of equipment is around there. But, you know, IV devices, 65% we found around the sink splash zone. You know, personal care items was 68%. You look at like alcohol, hand rub, and your PPE, 57% of that's within your splash zone. You know, H was like care devices in contact with skin. So lots of things that can be in contact with the patient. Ali's like crudely done a bit more work on it, looking to say what could be associated with bloodstream infections, what could be associated with VAPs, things like that, and colonization. So she's grouped it with different colors, as you can see. So there's lots of 1, 2, 3, 4, 5, 6 like groups of equipment that you could associate with bloodstream infections and then colonizations and VAPs. So you can see how patients could get Pseudomonas or any kind of bugs on them. So that, in essence, was the paper. A very simple study, but just showed you what patient equipment could be around that sink splash zone and how far water could splash. So that water could splash from this. You know, if it had Pseudomonas in the water, could easily splash onto a patient. And we could see that's how we might get transmission from the sink. So we've done a bit of further work on this sink splash zone, and hopefully this might be published in the next few weeks anyway. So this is just hopefully you can see that picture as well. That's just a hand sampler, sorry, not a hand sampler, an air sampler that you put an agar plate on the top of it. You let it run for 2 minutes and see what you can sample from the environment. So what we did was we actually ran a tap and put this sampler right next to it. And these are the plates from this sampling, from this air sampler that we got. And trust me, that you've got fungi on there, you've got gram negatives on there, you've got, you know, ASBs, aerobic spore-forming bacilli, things like that. We've done a bit more work and tried to characterize some of these bugs, and you get a whole range of bugs from splashed from this sink. So we've got some gram negatives such as Citrobacter, Enterobacter, Pseudomonas, Sphingobacterium, loads of different things. And scarily, what we found down this down the sink as well, we found our own published this year about a hospital outbreak, a specific plasmid CPE outbreak of a KPC at our hospital, and we actually can find that down the sink as well. So it's quite scary what you can pick up from this air sampling. So now we've got direct proof that bugs can splash from the sink into the environment, into those like pieces of equipment around there, to the patients. There's direct evidence of that. And Amy Mathers has done lots of stuff on that in the US as well. Really interestingly, the TVZ, I put TVCs on here, but the total viable counts actually were a little bit lower when we had filters on, but when we removed the filters off the taps, the TVCs were actually, we got more splashing and we got higher TVCs, so high number of organisms. And then when you did hand hygiene as well in that environment, you got even more higher TVCs and hand hygiene. So you can see what kind of activities you do to a sink and tap, you can get more splashing of these organisms and pick up more organisms of that. And finally, just the final slide really was just final couple of slides was just to think a bit more like there's lots of talk about like a waterless ITU or waterless environment. Waterless, I don't really like the term waterless environment, but trying to use less water. But there's loads of studies out there looking at how like sinks on an ITU are associated with more infections. And used Hopman, you may remember this paper where they did like a series of waterless interventions and they saw a reduction down in their gram-negative colonization and bacteremias. So that's all I wanted to say. So hopefully it's given you a little history of why I've looked around Pseudomonas, why that's led me onto the sink splash zones, and then it probably open for discussion for me. I also did a podcast on it, so that's also interesting, listening to Martin Kiernan as well. So thank you, and I'll leave it to you guys to for a bit of debate.
Okay, thanks, Mark. That was great. Really fascinating to hear kind of the backstory of the paper, what took you along that path of thought and where it's taking you now. So that's really great. Okay, so it's over to you now. Let's get some questions flowing into the Q&A box. Just a quick one from me. That plasmid outbreak that you mentioned with CPE, was that polyclonal? So it was different clonal backgrounds, but the same carbapenemase gene?
Yeah, it's fascinating, really. So what, I don't know how, well, it's probably got in to the hospital somehow from like, you know, so we've obviously had a patient at some point that's come into the hospital with a KPC, but that KPC is fascinating in as much as it's evolved through time and in different departments as well. So we've done a lot of work with the university now looking at this, you know, of that KPC, and like specifically in liver. I say liver because that's where we've looked at it in more detail, but it's evolved over time. So what's it's developed is like managed to get insertion sequences in it, and it's able to suck in other resistance genes. What's quite scary when you look at it, we found like mercury resistance in it. We found like a chlorate dismutase, like a chlorate dismutase resistance gene in there. All kinds of different genes in there, and you can see this common backbone in all of them, and it's just evolved. I think, you know, I think you've probably seen something similar when you were at in your hospitals down in London as well, and obviously Germany have reported something similar. But it's like it's evolved over time. What we find is why we called it a plasmid mediated outbreak, because we find it in all kinds of different species. So when I found it down the sink, it was actually in a Citrobacter, and then we have, you know, it's all, so the plasmid's just jumping around everywhere really. What's quite scary, but I think that's probably what happens in most, you know, in other hospitals. We're like a tertiary referral, so we're massive users of like broad spectrum antimicrobials, what you would expect. You know, undoubtedly we could do a bit better and a bit more on that. But that will be a selection factor on that. But the KPC is fascinating. You can just find it in all kinds of different wards, all kinds of different departments. I've now found it down the sink. You know, it's all kinds of different, you know, it just evolves over time.
Interesting. It's very difficult to think how we're going to control that spread, but that's for another day. So let's take some questions to you now, Mark. So just to comment on the slow-mo water drop, that was amazing. I've not seen that before. It's incredibly telling and really, really powerful. So thanks for sharing that. So the first question is from Abaro, and it is, where was the Pseudomonas isolated from the sink? As in exactly where? Was it the sink tap head, the sink basin, the sink drain, the plug hole? And following on from that, so where and how should we actually sample sinks and drains?
Yeah, so the study at the beginning, when I looked at Pseudomonas aeruginosa like transmission. That was literally just water out of the tap. That's all we took, and we obviously found it in the water. When we disassembled, we were taking a tap off, and when you have Pseudomonas, when you look for Pseudomonas, you pretty much find it everywhere. You find it everywhere in like the TMVs. You find it all everywhere in it. Really, we did lots of other pieces of work to see if it was mainly through the, did we get it through the water system? So we took piping out at the back, but we could never find the Pseudomonas in there. So I think it's all colonized into the taps. What's interesting with that whole genome sequencing that we did, we saw a complete variety of different Pseudomonas aeruginosa isolates. It just wasn't one that we found across all the, we found like that dendrogram I showed was lots of different strains. So there's numerous reasons how we've got Pseudomonas in our taps. One, because the taps are probably not fit for purpose, because they're like these swan-necked ones. They're not now because we've removed them all, but they probably could have got contaminated at source. They could have got contaminated with like wastewater down the sink, all kinds of different, how we've cleaned it, all kinds of different things. But in that original study, it was basically water from the tap, basically looking at Pseudomonas in that one file sweep. Going to the sink splash zone where we found Pseudomonas aeruginosa, it's probably, it could, that tap, we've taken water samples out of that now. We can't find Pseudomonas aeruginosa in it, but it could easily be from the tap, or it could be from the sink and splash zone. That's something slightly different. But when you got, when we look at those original pieces of work, the Pseudomonas aeruginosa was like predominantly in the taps themselves.
Thanks, Mark. I had a question, but I'm going to link it to one of the ones that's come into the Q&A. So my question was, based on that work that you did with the splash zone and all the equipment that you found in that splash zone, has that led to a change in practice? And also then the question from Alexandra is whether or not you know of any ways or plans to amend the HTMs to accommodate for the splash zone.
Yeah, so interesting around the HTMs, I was contacted, it was you guys, it wasn't you guys, I was contacted the other week about the sink splash zone, and they're writing some new guidance, and they want me to, they've asked for all my work on the sink splash zone to encompass that in. So that's a watch and wait, and I've said I'm happy to be involved in that. I'm also on the HIS working party guidance for water as well, so no doubt it will be in there. I find, and going back to that's number one, the HTMs. Your second question, Phil, we haven't done huge amounts of work around looking at the equipment around the sink splash zone, but what we have done now, Alison is doing a course, so she's going to revisit that type of work again, and we're going to look at like putting specific interventions in. So we did a bit of education at the beginning, but what you find with education, I don't know if you find the same, John, with yours, but I find education drops off after about 3 months, and people tend to revert back to the same sort of practice that they do. You have we've got 25,000 staff in our hospital with a 10% turnover, so education works for a bit, drops everything down, and then it reoccurs, so you need constant education. But anyway, Ali has got she's doing like a research project, she's doing like a master's, and she's revisiting all this type of work again. So she's going to look more into why the staff are doing the practice that they're doing, and then we're going to put certain interventions in place again to see if we can really tackle that. I think if I was looking at it, I probably have less sinks on my ITU because I wouldn't have 231 tap outlets on my ITU when it's 100 bed. There's lots of different with the new hospital builds. You won't see as many outlets. What's a complete change of thought? But yes, that's a wait and watch really. So we're going to do a lot of work on that. So Alison's already started it. We've got university students going in and auditing all the practice on our ITUs, and then we're going to put specific interventions, maybe target higher risk ones, things like that, and see what makes a difference.
Okay, thanks, Mark. In terms of education, I think you said education drops off after about 3 months. I think in my experience, it's more like 3 minutes, maybe even 3 seconds from time to time, depending on the issue. Yeah. So we have to continually keep re-educating. It's like painting the fourth bridge. It's something we just need to keep going with. So a linked question to that idea of your 100-bed unit with 200 sinks. Tracy's just built a single room unit. I mean, not personally with her own hands, but has just been built. Do you think we've made the correct decision to have a sink in each patient room?
Yes. Now, when you go and have a look at our ITU, I don't know how it's described, but our hand wash basins are behind the bed space, and it's literally impossible to go and, if you want to do a moment of hand hygiene, it's really difficult to walk past the patient and all the equipment on an ITU to go and have a look at go and do a moment like wash your hands in the sink. It's really impossible. Because I did all that water sampling, and this is a true story. When I was, I'm going to say, why am I talking about this story? But when I was going to do all the water sampling, I cracked my head open and I had to put stitches in my head because I was trying to get around the equipment, and like I just walked around and smashed my head and I was bleeding. So the ITU stitched me up while I was doing it. So if you think that's how tricky it is, no one's going to go and use those sinks. So why have we got them there? So I think we have made a bit of a, all for goodwill and intention. But I love Martin's analogy, what he used at IPS conference about the wastewater is of a hospital is like the bowel of a human. And I think we have probably gone the other way where we've had too many. So I think it needs properly, carefully reviewed. I also think, what else can we do with a tap and a sink? What type of taps we put in, what type of sinks can we get in? Can we get ones that don't splash as much, shark fins ones, and the angle of water, how it comes down. There's a lot more to consider around that as well. So I think we probably have gone the opposite.
Thanks, Mark. There's a lot of discussion going on in the chat, which is great to see. And there's some posts around updates to one of the HDMs for high risk areas as well. Shireen Mark asks, curious whether you're able to trace the source of the Enterobacteriales isolates you found in the air sampling. Was it from the sink drain or from the water?
Yeah, very good question. I would probably imagine it's from the sink itself. But we're going to do another piece of work again with the innovative. We applied for a bit of funding. Giving all my research ideas away here, but that's we just applied for some money from the school to look at the metagenomics of what's down the sink. So that's we're going to do whole genome sequencing of what that is down the sink to link it up, and we'll probably do something with the water as well to get a proper idea of all of that. So if I was guessing, I'd probably say it's all from the sink and the splash zone, especially I know for a fact that Citrobacter, the KPC, was down the sink because I literally went down there with a swab and found it myself. But it's really environmental sampling is really quite tricky to pick these type of things up. And what I would say is all those bugs I mentioned, like the Enterobacter, they're quite resistant. We did susceptibility testing on them. They're quite resistant. Weirdly, what Enterobacter is, but they were quite resistant. They were meropenem resistant, so I put them down the looked for CPs in there. They didn't have CP, the core CPs that we looked at. But yeah, it's I'd imagine it's from the sink. But
thanks, Mark.
Yeah, I mean, you wouldn't expect an environmental Enterobacteriaceae species to be, or Enterobacteriales to be that resistant, would you? So it suggests that it's come from in the hospital building. Interesting. Have you got a decent scar from the incident on the ICU with the sinks that they stitched up?
A little bit. You can see it on there if you look closely. So I have.
It's not kind of Harry Potter-esque, but I think I can just about see it if I squint. Okay, next question from Sally. What challenges did you encounter when trying to implement recommendations from this research, and did it prompt any changes to your clinical practice?
Yeah, I think the ITU are really our ITU are really engaged. So I was up there the other day for a complete different reason, and I had one of the nurses come up to me and said, "When are we going to do a proper waterless ITU?" So I think they're fully engaged in it. I've done a lot of teaching around that to them, and the work that we've done. So I think people are on board with it now. They were very on board with the changing of the taps and that kind of stuff. It's a lot more tricky because we're where I'm trying to implement this piece of work is in a PFI, so it's a lot more tricky to like remove sinks and things like. I don't know if anyone's worked in a PFI before, but you know, you rent the building, it's quite tricky to do certain types of work. But I think the staff are quite like they are engaged to do that kind of work, and I you know, the work that we're going to do, they're looking forward to it. So it just, what I'd like to do is like a proper RCT on it all. I know RCT is a favorite thing, but I'd like to do some proper research work on it instead of just doing little snapshots. I always find with little snapshots, they're great for a beginning, and like you say, John, the education goes off. So I think we need to do something, you know, take that human factors off and do. We've got like a perfect situation where I've got 4 ITUs basically, so I can compare and contrast really what we do. So I think it needs hence it's Ali's research project for the next 6 months, year, to get all that kind of data, get all the metagenomics, then we can get something a bit bigger to really tackle it and look at it. I think it'd be an exciting piece of work.
Thanks, Mark. One of the things I'm really interested in is, you know, all the different interventions that people are considering, you know, when it comes to trying to reduce the transmission risk from sinks to patients. And you know, water light or waterless care seems to be attracting a lot of attention at the moment. But what about where those sinks are going to stay and there's potentially products that are designed to disinfect, you know, the drain? So there's a question in the chat about whether or not you'd evaluated them personally and had any success. But I guess my follow-on question from that is it similar to education in the sense that as soon as you stop disinfecting it, the problem is just going to come back because these organisms are going to be introduced?
Yeah, biofilm grows really quickly, as you know. You know, it's like it grows like a like a millimeter or whatever every, like, you know, like so many over like hours or what have you.
The E. coli one was an inch a day.
Yeah, there you go. Like
Martin's
something
paper,
of an inch a day, so that, you know, you can put stuff down the sink, and you know, there are different things that you can put down there, and they, you know, when you look at them, they do make you know, they do get rid of a little bit of, you know, the biofilm. You know, I've seen that myself. You know, some of the stuff, you know, when you look at like granules and what have you, they just sink down there. You probably need something that foams up, and the technology with there, you can get water, you can get like cone traps that can heat, you can get UV. There's different things that you can do, and we need evidence around that. There's like, somebody approached me the other day with like a puricator, it's like hot steam that goes down it and things like that to get rid of the biofilm. But I do think you know, when you're never going to get rid of biofilm, and I think the sink and traps are a risk. And if we have things that we can aid us, that's great. But you still, you know, like I say, you're never going to get rid of the biofilm when it grows. But we need to be a little bit better from like what we do from a human factors point of view. What we stick down the sink, have probably less or more sinks that we're going to use properly for what we're going to use them for, and then look at like how we design stuff that prevents less splashes or a bit further away from the patient or something like that. It needs a proper look at it. So I don't really like the term, and I don't think used Hopman, waterless, because you're never going to get something that's waterless. And with the sustainability agendas as well, what's right, it needs to be like yeah, water light, or using water, you know, water safely on a you know, using less water basically is what I'm trying to say. Like that's probably the term I would use.
Yeah, I think, you know, all those measures in combination, a bit more of a holistic approach potentially is something that we should consider.
Yeah, I'd agree with that. It's a really difficult one, isn't it? Because you could say, well, we're not going to have any patients or sensitive patient equipment within the sink splash zone, but certainly on my units, you physically couldn't do that because you'd have to take away beds, and that would be a bigger risk. So I think we do have to think about sink management strategies, even if the long-term goal is to reduce the number of sinks we have. It is a really open and live question for me, is how can we address contamination of the sinks? But just on that point, though, I have definitely heard people say, and recently, well, contamination of sinks isn't very important because we are staffed to wash their hands. They have significant organisms on their hands. We want them to be down the sinks, and they will naturally contaminate the drains, because if you've ever seen a sink drain, it's covered in green goo. So is it a problem that we have contamination in the drains?
I agree with you, John. You would expect to see it down there. Like when we've gone down a drain and had a look at it next to a patient who's got a CPE, for example, they found the CPE down the drain. So again, you're gonna find it down there. But I think it's just the risk mitigation of what you do from like what is the risk literally from that to the patient. So there's a lot of people who have big outbreaks of CPs, and okay, there's a chunk of like colonization in the community, but actually there's like sometimes there's an underlying reservoir of what you can never really tackle. So I know numerous hospitals that have had outbreaks of the same thing like us, and they're thinking, well, where's that from? But there's undoubtedly more that we can do from from that kind of thinking about like our wastewater and our sinks and our splash zones and things like that. So I think you're right. I think you need you need them on the unit, but it's ways of like how we tackle it and think about them safely. Like you talked about earlier, Phil, there's going to be stuff that we can do to the sinks. There's going to be we can get ones that don't splash as much. There's going to be some kind of fancy design, and that's when working with industry, things like that we can come up. I know there's lots of people on the call, but there's certain ones on like I've noticed on working with like industry and that kind of stuff of solutions that we can get that make it easier for practitioners.
Okay, question from Brent then. Can you walk us through the sampling procedure for the drains?
Simply, the sampling procedure for the drains is like literally a swab down the drain. It's as simple as that. We've done different ones, so you can just get like a routine charcoal swab that you can go down and get like the green goo, as you say, on there. You can use polywipe sponges. We use it with an environmental with polywipe sponges on there as well. I do think going forward, it's like looking at the metagenomics of what you find down there. So you can do whole genome sequencing straight from that, get the DNA and go and do whole genome sequencing on that. So what I did from a sampling point of view is I was being biased because I was looking specifically because I wanted to see if I could find that specific PQEB KPC. So I put the swab or the polywipe sponge in a bit of TSB broth overnight to enrich it, and then I plated it out on specific media. So I put it onto like a McConkey plate media with meropenem. That's on my slide. One of that, not that one, but that's the type of media I use with meropenem in there, looking for it. So, but I always find with environmental swabbing, I don't particularly like it. And it's quite tricky because you get 101 different organisms on there. But I would always do like an enrichment broth and then plonk it onto I say plonk, that's my favorite word at the moment, but culture it onto a plate. But that's me being naughty because I'm looking for it, brought me back to my PhD days with making up McConkey plates with meropenem. So, but that's just me knowing how I could select it out that way. But that's how we did it basically in a nutshell.
Thanks, Mark. I've got one final question, I think, which is building on what you were talking there about metagenomics. My research background in the lab that I was working on, there was a lot of emphasis placed on using molecular techniques because you can never replicate the environment that these organisms grow in things like drains. You can never get the same conditions for culture, nutrients, etc. So we're seeing lots of people now doing the metagenomic analysis to investigate what's there. Do you think there's ever going to be a point in time where metagenomics is going to be used frequently to be able to assess maybe the risk of what those organisms are within those sinks in things like high dependency or intensive care units?
Yeah, I've always liked, I'm because of my background as a clinical scientist, I think, microbiology is a tricky one when you look at microbiology because we've got like cheap like I say reagents with agar plates, and you can grow that. So microbiology has never really gone down the route of like biochemistry with big machines, that kind of thing. It's quite like labor intensive. But when you look at like the price of agar plates and culturing and that kind of stuff, it's quite low. It's you know, it's low fruit. But the more I think about it, you know, like with viral, you know, how we go down the virus pathogen point of view, we've gone more of a molecular route. I do think there's a massive role for whole genome sequencing. The more I look at it, you know, I think it's a technology that we can utilize. So working closely with the universities, okay, it's mainly been research, but it's so valuable to understand your epidemiology of like your bugs, your CPs, and you know what's down your sinks, you know, to like clinical specimens, that kind of stuff. I think it just, you know, there's more of a role for me to have that available for you, because it's so powerful, really, so powerful and so quick. So I think, you know, it's just getting that technology at like a reasonable cost, practical size to use in a you know, a bacteria lab, basically.
Thanks a lot, Mark.
Okay, so one final question then, Mark, to put you on the spot. Given all we've heard and all the evidence that's out there, the evidence you've generated and the evidence others have as well, what do you think should be our priority action? What's the next step with contaminated drains in particular in clinical environments? Do we need to generate more data? Do we need to understand the issues more? Do we have a good enough understanding of the variety of potential solutions out there? Or is it time to take action and actually make some changes to our practice?
Yeah, I think, you know, there's like now people are getting more and more on board with it, you know, the new hospitals program, and we heard of like the recent IPS conference, you know, people are taking it on board now. And then in the new designs of hospitals, you know, we're certainly thinking of that kind of stuff, what's great. And I think, you know, practitioners themselves, when you talk about they've had big outbreaks, you know, you have to speak to other colleagues of ours, they've taken, there's already starting to take action. So I think, you know, the field of infection control, we are taking action around like going more to, like I say, waterless, but you thinking about water and the risks that it has. But undoubtedly there's going to be more, there needs to be more evidence and more research to help us inform those decisions. You know, there's loads of articles out there now showing that, you know, in an ITU, sinks and etc. are associated with healthcare associated infections, and we've seen that. There could be a few more products to help us, but I think they're coming along. You know, people are thinking about that now. There's more on the market now than there was 5 years ago, you know, from, you know, people contacting me. So I think it's just a constantly evolving piece of work. I do think we need one great big piece of research to actually prove it, so you can go to board, you know, this is what we see. These are the mechanisms, multiple different routes. This intervention makes a difference. And that's like the way forward we should be going. So evolving, really.
Okay, brilliant. Thank you, Mark. Thanks for joining us. I know you're crazy busy. Always got a million things going and lots of plates spinning. So we appreciate your time and all the work you're doing in this area. It's great. Thank you to everybody for joining us today. These journal clubs really work because of the audience and the participation we have. And all the questions that keep coming. We haven't even got through all the questions today, which I think shows what a topical area this is, with lots of questions outstanding. So you should, I hope, be able to see the schedule for upcoming journal clubs between now and the end of the year. So next week, it'll be me talking about Mpox Clade 1b. Is it the next pandemic? Plot spoiler, short answer, no. But there's a lot more to it than that. So if you can come along, and that's based on a paper that's just been published looking at some epidemiological parameters of Mpox. And you can see the rest of the schedule there till the end of the year. Just a reminder that if you want to be alerted when we schedule a new journal club, that all the information is there on the IPC Partners website, as well as the recordings on YouTube. And one final thing to say, which is thank you for coming, and we hope to see you next time. Thank you. Bye.